Hey, it's Mr. Shrum, and we are going to get into genes, which is the heredity code. We're going to ask some clarifying questions about the relationship between DNA and chromosomes, and what roles do they play in passing traits from parents to their offspring. For the warm-up, we're talking about the term gene expression. How do you think the term relates to the total number of genes a human inherits from his or her parents? And for the sample answer, we inherit half our genes from our mother and half from our father. These genes are located on homologous chromosomes, but not all genes will be expressed equally in a person. For example, a dominant gene could block the expression of a recessive gene. Gene therapy involves the transplantation of normal genes into a genome in place of missing or defective genes. Many scientists believe that gene therapy will lead to revolutionary new treatments for cancer. Why do you think gene therapy is such a promising field for this disease? And for the sample answer, we have there are certain genes that lead to an increased risk of cancer, such as breast cancer genes. Gene therapy might eventually help prevent cancer in people born with these genes. Also, cancer stems from mutation in normal cells. Let's see that. Gene therapy might help target or stop these cells from growing. All right. DNA replication. You may not realize it, but inside you there are trillions of tiny copy machines, specifically your cells. To pass on genetic information from parent to offspring or to grow new cells, a cell has to replicate its deoxyribonucleic acid, or DNA. DNA replication occurs near the end of interphase. So right there. And before cell division begins. This process is quite complex, but surprisingly, there are rarely any mistakes. DNA structure. In 1953, James Watson and Francis Crick, you hear Watson and Crick if you're going to study more about biology, but they proposed the molecular structure of DNA. And for a better understanding of DNA replication, let's look at some of the key features of the Watson-Crick model. A DNA molecule consists of two polynucleotide strands that are wound around each other to form a double helix or helical, this type of shape. A polynucleotide strand is composed of repeating nucleotide units. Each nucleotide comprises a 5-carbon sugar, which is called a deoxyribose, a phosphate group, and a nitrogenous base. The base can be either a purine, which is adenine and guanine, or a pyrimidine, cytosine and thymine. The phosphate group and hydrophilic sugar molecules face outward, while the purine or pyrimidine bases form the core. And you can see that here. So we have the sugar, the little phosphate, and the base in between. And these are color coordinated. So thymine and adenine go together, guanine and cytosine go together. The two polynucleotide strands are joined together by hydrogen bonds between the nitrogenous bases of the opposite strands. A purine always pairs with a pyrimidine and vice versa. So adenine A pairs with thymine T, so AT, by forming 2 hydrogen bonds. Guanine G pairs with cytosine, and that is formed by 3 hydrogen bonds. So AT, 2 hydrogen bonds, GC, 3 hydrogen bonds. This pairing is called complementary base pairing. In this kind of pairing, the base sequence of one strand determines the base sequence of the other strand. The 2 polynucleotide strands are anti-parallel, which means they run in opposite directions. Taking all of these features together, we can see that the DNA molecule resembles a ladder. So it looks like a twisting ladder. The base pairings are the steps, and the sugar phosphate molecules are the side support. So the green side supports are made of sugar and phosphate. The bases in between are these steps made of these nucleotide strands. Before we explore the DNA replication process, we need to understand DNA directionality. Recall that the 2 strands of the DNA molecule are anti-parallel. Let's explore what anti-parallel means. The building blocks of DNA, or the nucleotides, contain the deoxyribose sugar. The 5 carbons of the sugar are numbered in the clockwise direction. So the 3 prime, and you read that as 3 prime carbon, has an oxygen-hydrogen OH group, while the 5 prime carbon is outside the ring and is attached to the phosphate group. The adjacent nucleotides in a strand are linked by a phosphodiester bond between their 3 prime and 5 prime carbons. And in here, we have the 3 prime end, and here's the 5 prime end, and then we have the 3 prime end and the 5 prime end. So you see how they're flipped, right? Okay, and here's another look. 5 prime matches with the 3 prime. 3 prime matches up with the 5 prime. Yes. And that's what they mean by anti-parallel. In other words, each strand will have a free 5 prime carbon at one end and a free 3 prime carbon at the other end. They are called the 5 prime end and the 3 prime end. The 2 strands of the DNA molecule run in opposite directions. So one strand will have an orientation in the 5 prime to 3 prime direction, and the other will be in the 3 prime to 5 prime direction. Okay? One's going this way, the other one's going the other way. Anti-parallel. Now, here's the original template strand, and then we have the newly synthesized strand. Semi-conservative replication. Now that we've explored DNA structure, let's see how a DNA molecule replicates. Watson and Crick proposed a model for DNA replication called semi-conservative model of replication. According to this model, the strands separate during replication. Both strands separate during replication. Both strands then serve as a template for construction of 2 new complementary strands. So each new DNA molecule will conserve one old strand of the template and one new strand of DNA. That's why the replication process is called semi-conservative. Okay. Moving on, the replication process occurs in 3 steps. Let's explore the whole process of the semi-conservative DNA replication in detail. Unwinding. Replication begins by unwinding the 2 strands. This process involves breaking the hydrogen bonds between the nitrogen base of the 2 anti-parallel strands. An enzyme called DNA helicase performs the task of unwinding. As the DNA unwinds, a Y-shaped structure called the replication fork is formed. Proteins called single-strand binding proteins, which is SSB, associate with the separated strands. SSBs prevent the strands from snapping back to their helical structure. Also, unwinding can create tension in the DNA. The enzyme topoisomerase relieves this tension by creating small nicks in the DNA strands. The breaks in the strands are sealed later. Topoisomerase also allows the strands to rotate freely around each other to relieve tension. Base pairing. So after unwinding, the enzyme RNA primase synthesizes an RNA primer, a short segment of RNA about 5 to 10 nucleotides long. This RNA primer is complementary to the template DNA and attaches itself at the initiation point. This step is crucial because the replication enzyme DNA polymerase can add nucleotides only to the 3' end of a DNA strand. The RNA primer provides it with the 3' end to begin the process. The DNA polymerase now extends the primer by adding nucleotides that are complementary to the template strand. However, RNA differs from DNA. RNA contains uracil, while DNA contains thymine. Therefore, the RNA primer needs to be replaced to have a complete DNA molecule. To replace the RNA primers, the replication process follows a mechanism. We'll discuss that later in the replication process. As the polymerase is busy constructing the new strand, it encounters a new hurdle. DNA polymerase can add nucleotides only in the 5' to 3' direction. Now remember that the DNA strands are anti-parallel. One runs in the 5' to 3' direction, while the other runs in the 3' to 5' direction. So the extending process is different for the 2 templates. The new strand that runs in the 5' to 3' direction toward the replication fork is synthesized continuously by the polymerase and is called the leading strand. The other daughter strand, which runs in the 3' to 5' direction away from the fork, is synthesized differently. Because polymerase cannot synthesize in the 3' to 5' direction, RNA primase synthesizes short complementary RNA primers. DNA polymerase that just extends these primers in segments called the Okazaki fragments because the second strand is synthesized discontinuously in fragments. It lags and is referred to as the lagging strand. And you can see that in this graphic here. Joining. So the last stage of DNA replication is joining. And this stage revolves around cleanup work. DNA polymerase removes the RNA primers and replaces them with DNA nucleotides. DNA ligase seals the nicks present between the newly synthesized DNA after replacing RNA primers and the Okazaki fragments. Once the DNA is replicated, cell division can take place. Each daughter cell receives the same copy of the DNA with all the genes it needs. Genes are areas on DNA that code mostly for proteins. A few genes code for molecules that are involved in protein synthesis. Protein exists in almost every part of the body and is needed for vital body processes. Okay, so a checkpoint question. During DNA replication, several natural proofreading mechanisms are in place to prevent an error from occurring. However, errors can occur occasionally. And if the error is not fixed, it becomes a genetic mutation. How can a mutation affect protein synthesis by a cell? Do you think there are different consequences if it happens in a gamete cell, which is a sperm or egg, versus a non-gamete cell? And for the sample answer, genes are responsible for coding proteins, which are needed for all of our body functions and to build our body structures. So a mutation could affect the way a protein is made. If the error occurs in the gamete cell, it could lead to a mutation in a growing fetus. Because all cells in the body form from two fertilized gametes, this mutation may exist in all the cells of the developing baby. If it occurs in another cell, a skin cell, for example, then the mutation would be confined to the cells in the skin tissue. Prokaryote and eukaryote DNA replication. Let's see how the DNA replication process differs in prokaryotes and eukaryotes. The top gives us the prokaryote cell and the eukaryote cell. Cellular structures of prokaryotes and eukaryotes differ, and so does the complexity of the DNA molecule. These differences make the DNA replication process slightly different in the two types of cells. This table shows the main differences between prokaryotic and eukaryotic DNA replication. Prokaryotes, they have circular DNA. The DNA molecule has only one origin of the replication. They lack a nucleus, and the DNA replication takes place in the cytoplasm. Replication happens at only a single point in each chromosome. Eukaryotes, the DNA molecule unwinds at multiple locations. The DNA molecule has several origins of replication in each chromosome. DNA is contained in the nucleus, so DNA replication occurs in the nucleus. Each chromosome replicates as a section. Replication occurs simultaneously at several locations across the length of the chromosome. And I'll leave that here for today. And next time we'll pick up at the Mendel and gene expression points in this tutorial. But until then, have a great rest of your day/week, and I will see you all next time. Bye-bye.